Good morning and welcome to this IAS 2016 press conference to discuss progress and challenges in research to develop a cure or lasting remission for HIV infection. My name is Françoise Barré-Sinoussi and I am the co-chair of the IAS Towards an HIV Cure initiative. Last week, we released the International AIDS Society Global Scientific Strategy Towards an HIV Cure 2016. This strategy, published in the journal Nature Medicine, was developed through a 2-year process by 59 members of an IAS International Scientific Working Group. And this strategy was reviewed through an extensive global peer consultation process. The new Towards an HIV Cure strategy incorporates 4 years of scientific advances made since the publication of the previous IAS Global Scientific Strategy published in 2012, including a considerably more refined understanding of the nature of HIV research. HIV cure. Advocates play a central role in keeping all of us focused on the need for an HIV cure, in building support for cure research, and in ensuring that research is conducted ethically and with the expectation and needs of patients in mind. I'd also like to draw your attention to a new report on HIV cure resource tracking made by the IAS and IVAC. It shows that just over $200 million US dollar was invested in cure research in 2015, which million of US dollar invested in 2012. That trend need to continue. At the same time, we must emphasize that the funding for HIV cure research is not a substitute for full funding for treatment, care, and prevention. Today we are going to take a closer look at the science behind HIV cure research. I'm joined by my colleague, Sharon Lewin, by 4 other distinguished scientists who will give us a very brief overview of studies that presented at the Towards an HIV Cure Symposium, along with their perspective on those studies, tell us about the future direction in cure research. And in the room, we also other scientists who were presented at the symposium, so if you have any questions, they are also in the room. But first, I'd like to ask Professor Sharon Lewin to give us a brief overview of the IAS Global Scientific Strategy toward an HIV cure 2016. Sharon we are the triumvirate of the IAS HIV cure. She is also the director of the Peter Doherty Institute for Immunity and Infection at the University of Melbourne, and as I said, she's co-chairing with me and Steve the Towards an HIV Cure Symposium. Sharon?
Thank you, Françoise. Françoise has introduced the process by which we develop the strategy, and I thought I'd talk about two main things: the key achievements and changes since 2012, and the key priority areas going forward. So first of all, since 2012, there's been a really marked shift in the terminology we use around cure research and what we think might be possible. Back in 2012, we'd already heard about Timothy Brown, the only man who's truly cured of HIV, and that still remains the case 4 years later. What we now know, though, is that remission, meaning being able to stop antiretroviral treatment and remain well with the virus under control, the word that we use commonly in the English-speaking or the world, but perhaps is less commonly used in other cultures, is something that we're aiming for with these efforts. A true cure is an aspirational goal, and our intermediate goal is to achieve remission, allowing for people to stop antiretroviral therapy safely. So over the last 4 years, we've had numerous reports of this being possible, still very rare, but possible. I think over the last 4 years there's been an improved toolbox to measure HIV in people on antiretroviral therapy. We still don't have the perfect biomarkers to predict who can achieve remission, but we have a lot better tools to measure the virus both in blood and in tissue, but this is an area that still remains a major focus of the new strategy. We have much better animal models to study HIV cure, and you'll hear about that from Anne Cherudi later with advances with now paediatric animal models, a very important part of pushing the field forward. And finally, there have been a number of clinical trials over the last 4 years, primarily around latency reversing agents, that have told us that just shocking the virus or waking the virus up, what has been done on several clinical trials may not be sufficient, or is not sufficient to eliminate latently infected cells. So the top line messages from the strategy are that we still need a lot more basic research, a lot more understanding of where the virus persists, why it persists, and how to harness the immune response to eliminate it. We need, there's an increased focus on some key populations that may have an increased chance of achieving remission, and these include people treated very early in infection, who tend to have lower amounts of virus on treatment, or in the paediatric population who are also treated very early in infection and may have very different hiding spots for the virus because they have a different immune system. And I think the strategy is far more optimistic about two lines of interventions. That's around immunology and gene therapy, the advances in vaccines, the tremendous investment that's gone towards developing a protective vaccine may be very relevant for what is a therapeutic vaccine, or using a vaccine in someone already infected. And we heard about that at the symposium. And finally, some really exciting advances in gene therapy and new gene editing tools that can potentially remove the virus from cells. This is just happening in test tube models currently, or make cells resistant to HIV. And I'll again just emphasise the importance for the IAS on a cure being scalable, cheap and available to everyone, and therefore ethics, social research, engagement with community is a core part of the strategy as well. Thank you.
Thank you very much, Sharon. Now we will hear next from Anne-Marie Wincy of the University Medical Center in Utrecht in Netherlands. Anne-Marie presented at the symposium study on allogenic stem cell transplantation in HIV-infected individual. It's part of a consortium called the EpiStem Consortium. Anne-Marie?
Thank you, Françoise. So I think it's very important to realize that EpiStem is not a clinical trial, it's an observational project in patients who have a life-threatening hematological condition and therefore require a stem cell transplantation, and those patients have all consented to have large blood draws, really large blood draws, and if needed, tissue sampling to get insight in the effects of the stem cell transplantation on the viral reservoir. We have included 24 patients, of which 15 have been transplanted now from various countries all over Europe. And all of these patients, independent whether the cells they received at the stem cell transplantations had a genetic variation that prevented the CCR5 receptor that HIV uses as an entrance is there, or that they just had cells without or with this receptor, it doesn't really matter. All these patients, they have a decrease of the viral reservoir in a way that has not been shown in any other cure strategy. This is durable during still use of antiretroviral therapy. So none of the patients have interrupted antiretroviral therapy. Then we have 3 patients who have been transplanted more than 3 years ago. 2 of these patients are from Madrid, from the group of DS Martin, and 1 patient is from Düsseldorf, from the group of Professor Hausner. And only the patient from Düsseldorf has had the same kind of procedure as the Berlin patients. So he receives stem cells with a genetic defect that do not allow entrance of HIV. The other 2 patients from Madrid both received stem cells that are still capable of being infected with HIV. Then these 3 patients have been followed over time, and 2 of these patients, one of the patients from Madrid and the Düsseldorf patient, had serious graft versus host disease. That means that the stem cells that they received have attacked the immune cells from the patients themselves that are still present. And apparently, in these 2 patients, we cannot find any viable HIV in the blood. In the Düsseldorf patient, who had the same procedure as the Berlin patient, we can only find a trace of HIV if we do multiple tests in the tissues. You have to think then about the intestines, the ileum, and in subsets of cells in the blood. Interestingly, the 1 patient from Madrid that also got graft versus host disease and got normal cells with stem cell transplantation. In this patient, we cannot detect any HIV in the tissues anymore. So the conclusion is that independent of the cells that were used during the stem cell transplantation, we see an enormous reduction of the viral reservoir. And 2 patients we can not or hardly find any HIV. We don't know whether these patients are cured because they're still on therapy.
Thank you. Thank you very much, Anne-Marie. Now we turn to the only man of our panel today, indicating that pure research is involving a lot of female scientists, which is really great for me. So the next will be Dr. Giorgio Botti of the US National Cancer Institute. Giorgio will describe the key findings and implications of the work he presented at the symposium, indicating that there is no evidence of ongoing replication in tissue compartment during a combination antiretroviral therapy. Giorgio?
Hello? Okay. So, apologies for being a man, first of all.
We need some.
Our lab in Frederick is interested interested in persistence, like I guess everyone else in the room. And the way we approached it was the work I presented is about we studied HIV dynamics occurring in tissues in long-term CAR-suppressed patients. So we were aiming at using genetic analyses on single genome sequences to detect cryptic cycles of ongoing replication in those tissues that are traditionally labeled as sanctuaries. So to do so, we studied gout into healthy patients undergoing colonoscopy and multiple tissues obtained at autopsy in a third individual who had disease due to health complications. And the point is, our cohort was composed of people who was treated early and had sustained CAR suppression from 8 to 16 years up to the moment of tissue collection. And we didn't detect any sign of accumulation of genetic diversity, and therefore we concluded there was no ongoing replication in this specific set of early treated, long-term suppressed, but very different individuals. And as a side observation, we detected in 2 out of 3 patients cellular clone that persisted throughout the years, and once again, it was not anatomically restricted, and we think that this might be something that needs to be addressed in future cure strategies. So no ongoing replication either through time or tissue anatomical sites.
Thank you, Giorgio. Next will be Anne Chaudi of the Emory University School of Medicine, and will say a few words about the study she presented also on SIV persistance in antiretroviral therapy-treated infant rhesus macaque. So one presentation of animal model, the presentation of animal model after.
Thank you very much, Françoise. I'm happy to discuss my work to develop a model of simian immunodeficiency virus infection and antiretroviral therapy in infant rhesus macaques. And this work was conducted at the Yerkes National Primate Research Center and Emory University in Atlanta, Georgia, with collaborators from the University of North Carolina, the Center for Infectious Disease Research, and the NIH. And I'll share with you 4 major conclusions from our study. We found that antiretroviral therapy was well tolerated and effective in suppressing viremia in infant monkeys. We found that with ART, SIV levels decreased in the peripheral blood and lymphoid tissues of these monkeys, but the levels of SIV RNA remained elevated in the gut and the brain. We also observed heterogeneous antiretroviral drug levels in the lymphoid tissues and in the colon, but we observed very little drug present in the brain. And we found that naive and central memory T cells appear to constitute a major reservoir in SIV-infected ART-suppressed infant macaques. So this model provides a novel in vivo platform to test cure or remission approaches in infants. And we think that moving forward, we can use this model to assess different ways that we may be able to promote remission or test therapeutic vaccines or monoclonal antibodies or a number of other strategies. Thank you.
Thank you very much. And now we will conclude with Dr. Barbara Felber, also of the U.S. National Cancer Institute. Barbara will briefly discuss the presentation also she made on heterodimeric IL-15 induces effector cells activation and trafficking in the germinal centers of SIV-infected macaque. Barbara.
Thank you very much, Françoise. So IL-15 is an essential cytokine for the homeostasis of the innate and the adaptive immune system. George Pavlakis and myself discovered that IL-15 actually is a heterodimeric cytokine. That means it consists of 2 chains. And this allowed the production of these cytokines from mammalian cells, which is essential because it's a highly glycosylated protein. And we achieved something no biotech company has been achieved before because it required the recognition of this to be a heterodimeric protein. IL-15 is now acquired by Novartis and is being tested at the National Cancer Institute for cancer immunotherapy. IL-15, we believe, is a beautiful molecule that bridges cancer immunotherapy and HIV immunotherapy. And why is this? Because we found and reported here using different methodologies such as flow and imaging that with IL-15 you produce more cytotoxic CD8 cells. These are the cells that kill the infected cells, and more natural killer cells. Not only do we produce more of them, they go to the lymph nodes, and not only to the lymph nodes, they go to the germinal centers, which are known to be reservoirs of HIV. So, treating with IL-15, we can induce an army that can go to the right place to kill the virus. But we believe that IL-15 may need another help, which would be the induction of a supercharged army. That means soldiers that know exactly what to kill. And we believe that the DNA vaccine that targets highly conserved regions that we believe are the Achilles heel of the virus are important for that. And together with Jim Mullins of the University of Washington in Seattle, George Pavlakis and myself developed such a vaccine. And we believe that maybe together such a vaccine, together with IL-15 treatment, may be essential. Currently we are testing all model systems in rhesus macaques. So these studies are not yet in humans. And we hope we learn from the macaques so that we can extrapolate for human trials. Thank you.
Thank you very much. So now we will open the floor for question and discussion. I think the panel has been covering quite well different aspects that was mentioned before by Sharon around the priority of HIV cure research. And now please go ahead for question.
Hi, my name is Jürgen from DPA, the German News Agency. I have a question for you, Madame Marie-Sinoussi and Sharon Lewin. You both said that a cure is an aspirational goal and that it is not a replacement for the current efforts that need to be funded. So I'm wondering, with your experience, is there any way to guess whether we're way a decade or whether it might be 20 years, or is there no way at all to say that when there might be a cure? Thank you.
Sharon will say a few words also. I used to really answer, we don't know when we will have a cure and if we will have a cure. That is impossible to answer to this question today, according to the progress which has been made. We are very much, we really believe that we will be able to have a sustainable remission, for sure, when I don't know result. And that does not mean that we should not continue to try to develop novel strategy as well for cure. It's not because today we are not pessimistic, but we are more optimistic for remission. Is not also to say that a cure is impossible. It's just to say that to have a real cure will be very, very hard. That's all. But we have to try both, of course. And when, I don't know.
I totally agree with Françoise. I also just want to make a point that this is actually a pretty young area of research, although this is our second strategy. If you look at the investment in cure research, and it is increasing, it still is a tiny amount compared to the overall investment in HIV research globally and an investment in treatment as prevention. And as Tony Fauci said at an earlier press conference, We think that the current investment in cure is around in the hundreds of millions. The investment in treatment and prevention, which is 100% we know works and needs to get out there, is in the range of billions and billions of dollars. So it's a very early area. I think there has been quite a significant amount of progress, but the end goal of having a intervention that we know can induce remission, we don't know when that will happen.
Michael Smith, MedPage Today. A couple of questions, if I may, from Dr. Chaudhry. What's the clinical course of SIV in macaques, and we've had macaque research for some time in HIV, it seems to me. So what's new here? Why is this a new thing?
Thank you for that question. The novel aspect of what I'm presenting is that we're studying this in infant rhesus macaques. So there's been a number of studies, and Barbara has been involved in many of them to study SIV infection in adult monkeys. But because of the current situation with still 150,000 new pediatric infections every year and 2 million infected children, we think it's important to also model that infection and study cure strategies in infant monkeys. And there's a lot of differences between the adult immune system and the newborn or pediatric immune system, so there may be some differences in terms of the approaches we need to take.
A couple of questions about, this is an interesting area. You haven't done a structured TI in these patients yet, so you don't know whether there will be rebound. My question is, looking back at the Boston patients and Timothy Brown, you seem to be saying that serious graft versus host disease has some effect. And I don't know about Timothy Brown, but it seems to me that the patients in Boston also had some serious GVHD. So what's your thought on what's going on here?
Timothy indeed also had serious graft versus host disease. So that's indeed in all these patients. And I think the important thing here is what we see, that we also looked into tissues, which is something new. And also in Timothy, there was a trace of HIV detected, although we have to realize he was off therapy. And these patients are still on therapy. And I think that's our differences between the earlier reports and these patients. I think it's very difficult to say in 1 or 2 patients what would really be the cause. I think it's very interesting that we don't see a difference between the patients who had normal cells and the ones that had the genetic variation. And I think what is very important, and that's also why we bring this project here, is that we need to learn and we have to collaborate, and we really ask clinicians worldwide to bring in their patients who need this transplantation, so we can study them in the same manner with the most sensitive techniques of today to control.
Can I just make one point? Graft versus host disease is actually quite common after any allogeneic transplant. So to prove cause and effect, I think, is quite difficult because a lot of patients get it. One of the Boston patients did have tissue sampled and had no detectable HIV, as did 2 patients from Sydney that were reported in the Melbourne conference. So I think that, I'm sure Anne-Marie will agree, it's the treatment interruption that will tell us about cure, which is critical but a very difficult decision to make around the implications for patients.
Fred Schuck, FRTV, Portland, Oregon over here. This question is for Barbara Felber, or any of you if you wish to interject. The question is, we obviously know at some point it seems like there'd be multifactorial, multi-treatments to take to get to where we figure a cure would happen. In that light, is there a process by which you're either been doing or will change or that'll evolve over time to make decisions on what those compounds or modalities might be? As we go on, do we learn more about how we would, they're obvious when we bring the virus out of the cell and we kill it, but is there other things that will lend us to a better way of processing to multi-treatments that will get to a cure?
Yeah, I think in this context, the animal model is critical and will guide us. So it's going to be a combination between, in my case, interleukin-15 treatment. It's going to be what we learn from, because it's currently employed at the National Cancer Institute for cancer immunotherapy. So we are learning from that in humans. So we are learning from there in terms of dosing. For humans, we have been doing a lot of stuff in macaques, and we have been able to beautifully translate our data from macaques to humans. So we are now working on the infected macaque model system, and we believe that we can again then ultimately translate. This treatment that I'm talking about is not in place of antiretroviral therapy. It's going to be in addition, because antiretroviral therapy it has one problem, that obviously the virus can still hide. And the key thing is going to be this procedure together with other procedures to make that we can reduce the virus burden, so that we can reduce the sanctuary, the virus that is hiding in these reservoirs and sanctuaries. And by doing so, reducing the chance of getting more resistant viruses. So it's going to be in addition to the other treatment, modeled in the macaque model system first.
Bruce Jansen, infectious disease practitioner, reported a question about Wednesday. Actually, two quick questions. So, did the patients who underwent stem cell transplantation, do they have a conventional indication for that, like leukemia? And what are the plans for the consortium going forward?
All the patients had either leukemia or lymphoma or hematological life-threatening conditions. And what we have done is we have currently we're doing extra tissue sampling. So in one of those patients we have done, that I explained, looked in fluid from the brain. We have looked in specific T cell subsets. So we are going really in depth. We're looking at what we haven't done. We're planning is lymph nodes. And then I think it's very much up to discussion. At a certain time point, our tests are not more sensitive. And we really have to discuss with the patients and the ethical boards and their treating physicians whether we would continue therapy. I think in the end it's a decision of the patient himself, but it should be very well counseled, because we know it can have specific medical implications, but also psychological implications.
Dominique Tour from Le Monde in France. A simple question. Are we sure we have discovered all reservoirs so far, or is there any possibility that there are some undiscovered yet?
Maybe it's a question also for Georgie.
Well, I don't know if I'm the right person to answer to that, but I think maybe there's just, it depends on, I think, on what you think as a reservoir. Are you talking about tissue sanctuaries? I mean, I think there's just one, and these works are all approaching it from a different angle, but yeah, I think there's one, and it needs to be worked on more, but not that there's something that's still, not that there's a different hiding place, but just that the techniques to approach the way HIV persists needs to be worked on.
If I may add something to this, I think it's really important that again we are dissecting the animal model system for this, because in humans you can do, you don't have access to all these different tissues. In the infected rhesus macaque, however, we have it. And we are developing more and more and better technologies that we can understand what is the difference between the cells in different lymph nodes and different types of germinal centers. And I think we are all at the beginning of this. But the human is not the model system that you can use for this. but we are taking again our guides from the macaque to understand this. I think that's the best system.
Yeah, I think it's an excellent question, and I think I'm sure we haven't nailed that. Actually, one of the great exciting things from Giorgio's presentation was that he could look at autopsy specimens. So in a few situations we have the advantage of looking at tissue in humans. And the other area that's highlighted actually in the strategy that I think could play an important role is imaging. So new ways of PET scanning or labelling antibodies that can bind to a virally infected cell has already been done in monkeys. It was published a couple of years ago from memory, and those studies are actually now happening in humans, not yet presented, and that will give you a total body snapshot, which is much better than what we're doing currently, where we're trying to just target tiny bits of tissue from a very large pool.
Any other question? Yeah.
Just one small remark on that. The key question for me is not where do you find DNA, of course. It's more important to define where is the rebounding virus coming from. And to do that, you basically need to link indeed the snapshots sampling with the rebounding virus. And that's something that really needs to be still done. And that might be more important than finding traces of DNA which we don't really know. These are really indication-competent viruses. So we lack both technologies and good samples. And one of the technologies that I'm still looking very much forward to is this HIV full genome sequencing, to look if you have really intact virus that can give a replication-competent virus on a single cell level, which has not, been even broadly introduced in the research setting. So to answer that question correctly, the clinical setup, the animal models, and the technology, it will come to us over the next 2 to 3 years, I'm quite sure of that. But we really look forward to that data.
Thank you. One more question. If no more question, we will stop there. And again, I thank you very much for being with us today. And thanks to all the panelists. Thank you.